Let us start today's lecture. At the very beginning, uh, let me give a brief about the previous lecture. In previous lectures, we have discussed about the passive transport process. There are a number of passive transport process. We have discussed about the simple diffusion, facilitated diffusion, and osmosis. We have used some examples to express all those different type of diffusions like simple, facilitated, and osmosis. So in today's lectures, we will be focusing mainly on the active transport process. Now, what do we mean by active transport process and how this active transport process is different from passive? There are certain criteria based on which we can differentiate the passive transport from active transport process. But both the transport mechanisms in a particular cells is very important, right? Like in case of passive transport process, what we have seen, there is no involvement of energy, right? And another important features was in passive transport, the small molecules easily can pass through the cell membrane according to their concentration gradient or along the concentration gradient. And at the same time, what we have seen, there are numbers of channel proteins that also involves with the passive transport process. But here, the active transport is a little different. Like you can see here, it is a journey, right? So for this journey, like this is called an uphill journey. So for the uphill journey, the most important requirement is energy, right? So the process of active transport is also like a uphill journey. Energy is the most important requirement, and it occurs against the concentration gradient, right? So this is the most important features. It is an uphill journey. Energy is required. So the most important sources of that energy is the hydrolysis of ATP, right? And here the molecular movements occurs against the concentration gradient, right? No. So these are most important features of active transport. Like it is a carrier-mediated transport. Here, for the transport of a molecules, a large size molecules, it needs a certain types of carrier, and that carrier is very specific, right? So specific carrier, specific molecule can cross the cell membrane barrier from extracellular side to intracellular, as well as intracellular side to extracellular side, right? So carrier mediated transport, the most important features of active transport process. And in comparison to passive process, it is a little faster because it is carrier mediated. So sometimes cell needs certain types of molecules on an urgent basis, right? So to perform various types of cellular functions. So in that particular point of time, instead of using the passive transport process, cells can use the active transport process because it is a rapid process. And transport takes place against the electrochemical gradient, which is an uphill transport. It needs a certain types of energy, and this energy is basically utilized by the mediated protein or transport protein. And here another important features of that transport protein is it is ATPase transport. Means this transport protein has a specific catalytic property because yes, and molecules called ATPase is attached to the transport proteins. So this ATPase is an enzyme. So as it is the enzyme, it means it is capable to catalyze a specific type of reaction, right? Now it shows specificity. It means only specific molecules can bind with the specific type of protein, transport protein. The most important substances that are transported based on the active transport involved in the cell membranes are sodium ion, potassium ion, protons, chloride ions, and some other ions, along with some other substances like glucose and amino acids. So these are the most important types of ion and large size molecules which used for the transfer of that molecules from one side of the cell to the next. They use generally the active transport mechanism. You can see exact arrangements. So this is the cell membrane, right? So this is the transport proteins. If you see this transport protein, it has specific property, and in this transport protein contains ATPase attached to this, and this presence of that ATPase actually allows the molecular movements according to their concentrations. Mainly they have molecular movements occurs against the concentration gradient, means molecules moves from its lower concentration gradients towards higher concentration gradients by using specific type of energy called ATP. Here you can see in this particular case, the extracellular phase contains higher numbers of molecules in comparison to intracellular phase. Still, there is a movement of that molecules using this energy molecule from intracellular phase to the extracellular phase. So during the transport of that molecules, it shows some specific property called conformational changes, and that conformational changes of that transport protein actually allows the release of that molecules from one side to the next. Similarly, you also can see here in this case, concentrations of that molecules is less in extracellular phase, but it moves towards the opposite side, that is intracellular phase. So this is the general characteristic features of active transport process. Now let me explain in detail. Active transports can be classified into different types. Mainly the primary active transport and secondary active transport are the most important part of that transport process. Other than this, another important one is called endocytosis and exocytosis. Here endocytosis are two types, Pinocytosis and Phagocytosis. Now let me explain one by one by using specific type of example. So the active transport process, last time, okay? So this is the first one, the primary active transport process. So what is the features of that primary active transport process, and what are the molecules that are using that primary active transport mechanism? The most common example common examples of protein pump or pump that involves in the primary transport mechanisms are Sodium-Potassium ATPase pump or Sodium-Potassium pump, right? So here this Sodium-Potassium pump is very specific for sodium ion and potassium ion only, right? Similar to that, another examples of primary active transporter is calcium pump, and this calcium pump allows only calcium ion, not other ion. Similar to that, hydrogen ion or proton potassium pump, no, go outside, go. So proton potassium pump. So this is again another important pump that is used for the transfer of both proton and potassium ion across the cell membrane. Now, as I mentioned, that active transport requires a specific type of energy. So here, for this or for the actions of those pumps, they also require energy that is provided by the hydrolysis of ATP molecules, right? Now, during hydrolysis or due to the hydrolysis of ATP, it releases phosphate, right? and that phosphate actually act as a sources of that energy for the movements of molecules against the concentration gradient. See, so how this sodium-potassium pump actually act? This is the sodium-potassium pump called sodium-potassium exchange pump, right? And the specialty of that sodium-potassium exchange pump is it has some specific binding site for both the sodium and potassium ion. Now, if you see the properties of cells, in cell, there is a specific distributions of both the sodium ion and potassium ion in both extracellular phase and intracellular phase. Here, in this cytoplasmic phase called intracellular site, contains a more number of specific ion, mainly the sodium ion, right? Whereas in extracellular phase, there is a more number of potassium ion. But when the sodium-potassium pump act, so according to the phenomena, the molecular movements of us against their concentration gradient. So here, how actually it happens? You can see at the initial phase, 2 potassium ions that is attached to the outer side of this sodium-potassium exchange pump, because the outside of this pump contains the binding site for sodium potassium ion only, right? But 2 potassium ions can bind at a time. On the other hand, 3 sodium ions that can bind to the inner side, inner pocket of this exchange pump, right? So during sodium-potassium exchange, that both the sodium and potassium ion binds in specific positions of that pump. But here, the most important thing is, once these sodium ions initially binds to this inner membranes of that pump or inner side of that pump, there is an activation of the ATPase protein that is attached to that pump. So immediate after that activations, there is a conformational changes of that protein, right, or that exchange pump. So due to that conformational changes, it helps the release process of both the sodium ion as well as the potassium ion. Here, sodium ion moves towards its opposite side, like it moves, you can see here, it moves from intracellular side to the extracellular side, right? So here, the condition is, during this transport process, the numbers of sodium ions initially in that particular phase was less, right? So that though the concentrations of sodium ion in extracellular, in cytoplasmic or intracellular phase was less in comparison to outside, still there is a movements of sodium ion from intracellular or cytoplasmic phase to the extracellular phase, because according to the phenomena, the molecular movements occurs against their concentration gradient. So that actually helps in the efflux process of 3 sodium ion and influx of 2 potassium ion. This is just a general mechanisms how sodium-potassium pump or sodium-potassium exchange pump act as a primary active transporter, right? Here, due to this efflux of 3 sodium ion from cytoplasmic phase to the extracellular phase and influx of 2 potassium ion from extracellular phase to the cytoplasmic phase, how this distribution now affects the cell. See, it creates a high potassium ion concentrations and negativity inside the cell, because we know both the potassium ion and sodium ion carries the positive charges, but though they carries the positive charges, still what I have mentioned here, it creates high potassium ion concentration and negativity inside the cell. How it becomes negative? It becomes negative is because the number of positively charged sodium ion exit out is more than the influx of positively charged sodium ion. That makes a electrochemical gradient or electrical gradient. That is why the inside of the cells now becomes negative, right? And this distributions of ions or ionic distributions actually helps to maintain the cell volume. How? How it maintains the cell volume? As we have discussed in the osmosis process. In osmosis, what we have seen, solvent molecule moves from higher solvent concentration gradients towards lower solvent concentration gradient. It means, suppose in specific solutions where number of solute is more, it means it is concentrated, right? So here, due to this influx and efflux of both the sodium ion and potassium ion, actually it changes the osmolarity, right? And changes its osmolarity actually allows the cells either to gain more amount of solvent or lose more amount of solvent. That actually facilitates to changes in the cell volume. You can see from here how actually it maintains cell volume. Before that, you also can see how this ionic distribution is important for cell. This concentration gradient is essential for some specific function. The distributions of both the sodium ion and potassium ions takes place by using these transporters. How it is important for cell? Yes, we can see the importance of these distributions in nerve cell. It actually helps in the nerve impulse transmissions because ionic distributions, both the sodium and potassium ion distributions across the nerve cell membranes is very vital for impulse transmissions from one cells to others. At the same time, this distribution actually not only involved with the transmission of that impulse, by transmitting that impulse, it helps the nerve cells to become functional nerve cell. It also involved with the muscle contractions, and here this sodium potassium exchange pump is an important source of energy for another type of active transport mechanism that is called secondary active transport mechanisms. For example, in secondary active transport mechanisms, for the transport of both glucose and amino acids, they are not using energy directly. Instead, they are using energy from concentration gradient forms in primary active transport process. So, in that aspect, we could say the distributions of both the sodium ion and potassium ion, or the generation of concentration gradient, is very vital in active transport mechanisms like in secondary active transport process. How it maintains the cell volume? Suppose if sodium accumulates inside, water enters by osmosis, cause swelling. Yes, because accumulations of more and more number of sodium inside of the cell means it becomes more concentrated. So inside becomes more concentrated means it will allow more amount of solvent from outside to inside. So due to that solvent entry, there is a swelling of the cell. Pumping of sodium out prevents excess water entry, and by these mechanisms, it actually helps to maintain the proper cell volume. It creates negative membrane potentials because more positive ions go out than 2 potassium ion enters inside, and so in that distribution, both sodium and potassium ion distribution actually helps to maintain negativity across the cell membranes. So this is the sodium potassium exchange pump is important in functional point of view. Now there are certain molecules which can act as a regulator of sodium potassium pump activity. So what are those molecules that regulates the activity of the sodium potassium pump? You can see there is some hormones like thyroid hormones and insulin hormones. Both the hormones improves the activity of that sodium potassium exchange pump. So more active activity of that pump actually means that sufficient amount of sodium and potassium are distributed accordingly across the outside as well as the inside of the cell membrane. So it helps to maintain energy use as well as cell metabolism. Another important hormone is called aldosterone. It increases the number of pumps. Sometimes Sometimes cell may needs the release of more amount of sodium ion and influx of more amount of potassium ion, but the number of that pumps is less. So that less numbers of sodium potassium exchange pumps will not carry out the overall functional features of the cell. So in that aspect, cell needs a specific regulatory mechanism. Yes, there are certain molecules called hormones, and that molecules actually helps to improve the numbers of that sodium potassium exchange pump. By increasing that sodium potassium exchange pump, it helps to maintain the ionic balance accordingly. You can see some specific portions of the body like kidney. This helps in sodium reabsorptions and potassium excretions that helps to maintain the fluid and electrolyte balance. That is very common in kidney cell membrane. And there is some neurotransmitters also act as the regulators of that pump, other than certain types of drug, right? And both the neurotransmitters and drugs act as an inhibitors of that pump, right? So these inhibitions of the activity of those pumps is also important to maintain the balance. Increased numbers of sodiums inside the cells actually stimulates the pump to remove the sufficient amount of sodium ion from inside to outside. So this is what is the regulatory mechanism. These are the molecules which actively involved with the overall regulations of that primary active transporter called sodium potassium exchange pump regulator. Now this is again another important one, which is also a part of primary active transport mechanism called calcium pump. So as we know, the calcium is also very vital for cell, right? We can see the calcium is important for muscle contraction. Calcium is also important for the release of neurotransmitter from one nerve cells to the synaptic junction. So as cell needs a sufficient amount of calcium ion, so there must be specific transport mechanisms to maintain that sufficient amount of calcium ion. Now, how this calcium ion act here? It involves Uniport carrier, right? It means it only carries a single type of molecule, like here calcium. It helps to maintain the low calcium ion concentrations in intracellular phase of the cell. Now see, here this phase is called the cytoplasmic phase of the cell. This is extracellular phase. If you see the first part in the cytoplasmic phase or in the cytoplasm, the number of calcium ion is less, right? Whereas in this side the calcium level is high. So how actually it is possible? It is only possible by this calcium pump. And why this is important? Because as we know, for the better performance of cells, it needs to maintain lower level of calcium ion inside. So to maintain that lower level of calcium ion inside, this calcium pumps act as an important transporters, while the calcium ions can exit out from cytoplasmic phase to the extracellular phase. In extracellular phase, either it is transferred to the extracellular phase. Sometimes the calcium ions can be stored in the endoplasmic reticulum or sarcoplasms, right? So on an urgent basis, that stored calcium ion also can be used by cells. So in that conditions, here from extracellular phase, on an urgent conditions, cells can use the extracellular phase calcium ions for cellular function. So this is where calcium pump also important for the cellular functions. Now you can see where the calcium ion is most important, like as I mentioned in the muscles. When a muscle contracts, calcium is released into the cytoplasm, right? After contractions, the calcium pumps actively moves it back into the sarcoplasmic reticulum and leads to the relaxations of the muscles. This is an important phenomenon of the muscles. Once muscle contracts, it must return back to the relaxation phase. To maintain the contractile property and relaxations, the cell needs a sufficient amount of calcium ion, right? So to get that calcium ion, the transporter helps. In nerve cells, entry of calcium triggers the release of neurotransmitters. We have seen in nerve impulse transmissions, the release of neurotransmitter from synaptic vesicles is facilitated by that calcium ion. So how this calcium ion actually facilitated this transmitter release? If you see the structures of presynaptic area, presynaptic membrane contains this calcium pump actually, which is very specific. So during nerve impulse transmissions, that calcium pump which is present in the presynaptic membrane allows the calcium ion inside the presynaptic zone. So once calcium ion enters inside the presynaptic zones, it facilitates the vesicular movement towards the synaptic membrane, right? And ultimately helps the release of transmitter from that synaptic vesicle into the synaptic junction. So this is where the importance of calcium pump. So another important pump also acts in primary active transport like proton potassium pump. I have not mentioned here. Now comes to the second part called secondary active transport. So what is the basic difference between primary active transport and secondary active transport? Both are active transport process. In both the case, molecular movement occurs against the concentration gradient. Both are using energy, but the main difference is in case of primary active transport process, it uses the ATP directly, right? Whereas in secondary active transport, it also uses energy, but by indirect process. So what is the indirect process? It uses energy based on the concentration gradient formed in sodium-potassium exchange mechanism, right? And that concentration gradient actually acts as a sources of energy for the secondary active transport process. You can see here the most important properties are in this transport process. It is also known as coupled transport or co-transport. Coupled transport or co-transport means it carries in this particular transport mechanism, transporter carries two different type of molecules at a time, right? That is why it is called coupled or co-transport. There is no involvement of direct energy, but it depends upon the electrochemical potential difference that is created by pumping of ions in and out of the cells, mainly sodium-potassium pump. Now, this is very common example of secondary active transport process called sodium glucose transporter, right? This is the protein pump, right? This is the pump. This pump is also very specific. It only allows sodium ions and glucose ions, right? Now see how the sodium ion and glucose ions are transported across through this transporter. This is the transporter, right? Sodium glucose transporter present in the cell membrane structures. If you see this side of the cell, right? In this side there is a specific binding site, right? And that binding site actually consists of, that binding site is specific for both sodium ion and glucose molecule, right? Now, once this glucose and sodium binds to their specific site, there is a conformational changes of that sodium glucose exchanges, and that conformational changes actually allows the release, or it helps the release process of both the sodium ion and potassium ion accordingly. Now see how actually it happens. Here this sodium-potassium ATPase pumps that I have mentioned, that due to the concentration gradients, the energy is already formed, and this energy actually allows the journey in secondary active transport process called downhill journey. In previous case, what we have mentioned, this was the uphill journey, right? But here the terms mentioned, it is called downhill. What is the meaning of that? It means the molecular movements or ionic movements and the movement of substance are opposite to one another. Means in one case, what we see, there is an uphill journey, but in another case, there is a downhill journey. So in case of downhill journey, there is no involvement of energy, right? So here, these actions creates a downhill sodium ion gradient inside the cells, and this sodium glucose transporters uses that sodium ion gradients created by sodium-potassium ATPase pump to transport the glucose across the membrane. See here. In sodium-potassium exchange process, where the number of sodium ions was more, after exchange, outside of the cell, the number of sodium ions is more in comparison to inside, right? So here in secondary active transport, sodium ion moves from that extracellular phase to the intracellular phase, right? Means after the completions of primary active transport, the concentration of sodium ion is more in the outside of the cell, right, compared to inside. So in secondary active transport, along with the glucose molecule, sodium also transported, but where is what is the directions of the sodium movement? The directions of movement is from extracellular, where the number of sodium ion was more, towards the inside, where the number of sodium ion is less. So that is why it is called a downhill journey. Here, in secondary active transport process, downhill journey occurs for sodium ion. Clear? But the glucose ion also enters from outside to inside, right? From outside to inside. But here from one side to other, here the concentration is less from where glucose are entering towards its opposite side. Clear? Now see, this active transport process or the transporter that is involved in the active transport mechanisms are of different types. Some are called Symporter, Antiporter or Uniporter. The mechanism is called Symport, Antiport and Uniport, right? So what is the basic differences? You can see here, in case of Uniport mechanisms, this is the uniporter, means the pump, means the protein which allows the molecule. So these proteins only allows a single molecule at a time, called uniporter, right? Whereas you can see in case of Symporter, this is a protein called Symporter. The mechanism is called Symport mechanism. Here, two different types of molecules moves at a time towards its opposite direction, right? So, like here you can see, in case of Symport mechanisms, sometimes we can say the molecules may be similar in natures, right? The most important features is the directions of that molecules will be the same directions. Whereas in this case, this is the protein or this is the transporter called antiporter, and here's the molecular movements occurs in two different directions. So, like as we have already discussed that sodium-potassium exchange. So here, movement of sodium occurs from inside to outside, and for potassium from outside to inside. It means sodium-potassium exchanges act as an antiporter, right? Whereas sodium-glucose, just now we have discussed, both the molecules moves in the same directions, sodium-glucose transport, right? So though the molecules are of different types, but the directions of both the sodium and glucose in the same directions, right? It is a common example of Symporter. And whereas sometimes we can see the single molecules also can move, like we can see in the movements of glucose through GLUT, right? So glucose transporters, whereas only glucose molecules can move through these transport proteins in one direction. So this is what is the different types of transporters, and what is the mechanisms how these transporters are involved in the transport of different types of molecules. You can see here Symporter. So now next point. Here the next part of active transport mechanisms is called the endocytosis, right? So what is the basic phenomena of endocytosis, and what are the different types of endocytosis process? Yes, it is the process by which a cell takes in materials from outside by engulfing them, right? And the materials are of different types. Sometimes it may be the liquid materials, sometimes it may be the solid materials. The size of the materials may vary, right? The most important mechanisms of the endocytosis is engulfing of those materials from outside of the cells towards inside. Now, if you see the overall mechanisms of the endocytosis based on the mechanistic features, they are classified into different types like Pinocytosis, Phagocytosis, and receptor-mediated endocytosis. So the most important features of the Pinocytosis is actually it is non-specific, right? You can see this Pinocytosis a mechanism for all the different types of cells, right? And it can engulf any type of liquid materials. There is no specificity for the engulfing by this Pinocytosis mechanisms. This is also known as the cell drinking, right? Whereas phagocytosis is very specific. It can be found in some specific cell types only. Like you can see this some types of immunocells, right? So-called phagocytic cells. Here in this case, there is an involvement of a specific type of receptors that are present into the surface membranes of the cell, and that receptors actually specific for the substance which can bind to this, right? Now, see the general mechanism or overall mechanisms of the pinocytosis. As I mentioned, that it is nothing, it is also known as the cell drinking, right? And it is non-selective process, which is a continuous process that may be found in all the different types of cells. The most important properties of pinocytosis. Here, the importance of that pinocytosis is not only the engulfing of the materials. By engulfing the materials, it helps to maintain the balance of the cell membrane, right? You can see what is the exact mechanism. A membrane deforming protein attached to the membrane help to bend or carve the membranes inward to form a pouch. It means you can see there is a specific type of protein present in the membranes of the cell called membrane deforming protein. And how this membrane deforming proteins are important for pinocytosis? It actually helps in the formations of pouch, right? It helps in the formations of pouch, right? And during this pouch formations, another important type of molecules also played a role called dynamic. Here, the formations of the pouch by dippings of the membranes, there is a dippings of the membrane, and this dippings actually helps in the formations of a vesicle called endocytic vesicle. And that endocytic vesicles are nothing. These endocytic vesicles contains the fluid materials, which is extracellular fluid materials, right? Now, after the formations of that vesicles is pinched off by proteins. See, there is a role of that dynamic proteins that helps in the pinch off process, right? And ultimately, it releases inside the cell. Means these vesicles now release into the cytoplasmic phase of the cell. So, by this process, or by the process of pinocytosis, it helps removal of extra plasma membrane that was embedded during exocytosis. This is what is the importance of the process pinocytosis. As I've already mentioned, this pinocytosis process actually helps to maintain the balance of plasma membrane by engulfing certain types of liquid materials by the formations of a pouch. So you also can see another important one called receptor-mediated endocytosis. The most important features of that is there is a specific type of receptors present into the surface membrane, and once these specific molecules whose size is a little larger, which is not transported through the primary active transport process or secondary active transport process, so they can follow this, they can use this receptor-mediated endocytosis process. They bind to the specific receptors, right? And by the bindings of that, these molecules or extracellular molecules, they form a complex, and that complex and nothing, actually it is called as coated pit, right? And this coated pit contains a specific type of property, like it contains some, this coated pit contains some molecules which has some protein property, like it contains some actin fibers, right? So this endocytosis, actually, receptor-mediated endocytosis actually helps for the removal or killing of those substances which are harmful for the body. Like you can see, endocytosis of virus such as hepatitis H virus and sometimes some other molecules also can be endocytosed by this receptor-mediated endocytosis. And after the endocytosis process is over, or after the engulfing of all those molecules inside, then by the process of other biochemical mechanism, biochemical process, it is processed further. Whereas in case of phagocytosis, this is very common, as I mentioned, that this process are specific for some immunocells, right? It is a process of internalizations of large multimolecular particles, bacteria, and some dead tissue by specialized cells. For example, MBBS, right? And the materials makes contacts with the cell membranes, which then invaginous. This is a general mechanism of endocytosis, how the phagocytic cells works, how the immunocells works for the engulfing of foreign particles like virus and bacteria. So up to this, in today's lectures, what we have discussed? We have discussed mainly three different things: the primary active transport mechanisms and the role of different types of protein pump or carrier protein, like sodium-potassium exchange pump, calcium pump, proton-potassium exchange pump. And in secondary active transport, we have mainly focused on specific examples like sodium glucose transporter, which is co-transporters. And after that, we have discussed the different types of transport protein or transporters and what is the mechanisms. And finally, we have discussed in brief way what are the different types of endocytosis mechanisms like pinocytosis, phagocytosis, and receptor-mediated endocytosis. Thank you.